Very good evening, student. So we have completed the three lectures uh, till yet. So in the last lecture, we have done with the structure of anthropus ovule, the development of female gametophyte, and some part of this outbreeding devices. Okay, so again we're going to start with this outbreeding devices. The outbreeding devices are nothing but for the maintaining of the genetic diversity. Okay, so in your textbook they have given the 6 different types of outbreeding devices. So first is unisexuality, second is the dichogamy, third one is the prepotency, fourth one is heterostyle, fifth hercogamy, and last one is the self incompatibility. So all these six types are the outbreeding devices, that is nothing but the cross pollination. It comes under the cross pollination to maintain the genetic diversity. As we know that from asexual type of reproduction or self pollination, the genetic diversity can't be maintained. So it is only because of the cross pollination we can maintain, or the plant can maintain, the genetic diversity. Okay, first we'll start with the unisexuality, that is unisexual. Okay, that is nothing but the sexes are separated. Okay, that is what is known as what, dioecious plant. Okay, it is also called as what, unisexual plant. So in that the male or female flowers are different. Okay, so that is what is known as what, sexes are separated, and it comes under the unisexuality. Okay, second, so they have given the example for that is mulberry and papaya. Second outbreeding devices, they have given the dichogamy. In that the maturation of anther and stigma is at different times, so that it can follow the cross pollination. Okay, so they have given the two terminology in this case. First is the protandry and second one is protogyny. Protandry related to androecium, that is for maturation for anther. Protogyny, it is for gynoecium, that is maturation for stigma. Okay, so for protandry they have given the example in the disc florets of sunflower, and the protogyny they have given the example, that is gloriosia. Right, third type is prepotency. In that pollen of pollen of another flower germinate rapidly on the stigma than the other pollens. Okay, means what, suppose we can take the example, they have given the apple. If the apple flower, the same flowers pollen grains cannot germinate on the stigma than the another flower of apple, that is why it is known as what, prepotency, that is pollen of other flower from the same plant germinate rapidly on the stigma of the other stigma than the other pollen grains. Right, fourth one is what, heterostyle. Heterostyle, it is the heterostyle is also known as what, heteromorphy. Okay, so they have given the example of primula, that is primrose. In that there are 2 to 3 forms of flowers in which the stigma and anther are placed at different levels. Okay, if their position are different, so there is a less possibility of self pollination. Right, next is hercogamy. In hercogamy there are a physical barrier which prevent the self pollination. Okay, for example they have given the calotropis. Calotropis, अगर आपको पता होगा, रुई का पेड़ जो हनुमान को जो पत्ते चढ़ाते हैं, जिसका एक छोटा फूल रहता है, if you see in that the stigma is pentagular. Okay, so it is like that. Okay, so it is above from the anther, so that it will maintain the physical barrier, so that it will prevent the self pollination. Right, next type is what, Next type is self incompatibility, that is nothing but the self sterility, right? So in self incompatibility, it will inhibit the other pollen grains to germinate on the stigma. Okay, that is inhibition of germination of pollen on the stigma, and they have given the example of tobacco or thea, right? So these are what, these are the some outbreeding devices, some the type of outbreeding devices for maintaining the genetic diversity. Here we have completed the type, that is pollination type, that is cross and self pollination. Okay, next point of our discussion is nothing but the pistil pollen interaction. Okay, as we know that ki pollen are falls onto the stigma, and from there it starts its journey towards the egg for fertilization, right? If we you know that it comes under the sexual reproduction. So first they have given the pollen pistil interaction. Pollen, it is just for androecium. Pistil, it is, you know that it is for gynoecium. Okay, so pistil is what? It is a complete structure, that is stigma, style and ovary. Combinedly it is known as what? Pistil. Okay, so interaction of pollen grains with a sporophytic tissue. Sporophytic tissue is nothing but what? Stigma. It begins with the pollination and ends with the fertilization, is nothing but the pollen and pistil interaction, right? Now in this pollen pistil interaction, so many right type of pollens or wrong type of pollen fall onto the stigma surface. So there are specialized protein which recognizes this right type of pollens and discards the wrong type of pollen, right? So that is why they have given the two terminologies, that is compatibility and incompatibility. Okay, so in compatibility, it is the proteins which are guarantees the right type of pollens onto the surface of stigma, and incompatibility, it is nothing but a rejection of a wrong type of pollen grains through this special type of protein, right? So after falling onto the surface of stigma, these pollen, this right type of pollen grains absorb the water and nutrients which are present on the stigma of the flower, and they will develop the pollen tube. Okay, so for the development of pollen tube, they required water as well as nutrient, so that they can form the pollen tube and reach to the their target, that is nothing but the egg. After this, they have given the one term over there, that is artificial hybridization. If you want to improve variety of crop, then in a laboratory condition we can hybridize the plant with a correct or a right type of pollen grain with the flower, right? So next term is nothing but the double fertilization. Why it is known as double fertilization? Why we are calling it a double fertilization? Because in the double fertilization, in the double fertilization, the fusion of one male gamete with the egg and that of another male gamete with the secondary nucleus is there. That is why it is known as what? Double fertilization. Yeah, as we know that we have seen the development of male gametophyte. In that you have seen the one nucleus is there which is forming the tube, that is tube nucleus, and another one is the two male gametophyte is there which is following the tube nucleus. That comes into the embryo sac. The first nucleus fuses with the egg, and another nucleus is going to fuse with the secondary nuclei. That is why it is known as what? Double fertilization, because two male gamete fertilizes the two nuclei of embryo sac. So we are calling it as double fertilization, right? Here I have made the diagram. That is, this part is what? This is the part of stigma. this is the style, and this complete is the ovary. Stigma, style, and ovary, right? So this orange colors dot are nothing but what? These are the pollen grains, and they are forming the pollen tube. Okay, so I have shown the one pollen tube is germinating and other is reaching towards the embryo sac, right? Now these are the pollen, this is the pollen tube, this is ovule, this is embryo sac. You know that, you just recall it. This is a micropyle, this is the chaliza, these are the integuments in that. Here is the antipodals, these are the synergids with surrounded the egg cell, right? And this complete structure you know, right? Now what is happening in the double fertilization? Fusion of one male gamete with the egg and another with the secondary nucleus. Guidance to the pollen tube is by chemicals of synergids. Now they have given the three types of entries of this pollen tube to the embryo sac. Okay, first they have given the porogamy. Whatever I have shown over here is nothing but the porogamy. The pollen tubes comes or enters into the micropillar end is nothing but the porogamy, and this entry of this pollen tube into the embryo sac is totally depend upon to the chemicals which is secreted by the synergids. These are the synergids which is surrounded to the egg, right? So the first I have shown over here is nothing but the porogamy. The pollen tube enter through the micropillar end. Second is the chalizogamy. The pollen tube will enter into the chalizal end from here, right? And third one is the mesogamy. It will enter through the integument from here, or it may be from here. Okay, but ideal fertilization takes place through the micropillar end only, right? Next they have given that this pollen tube enters through the micropillar end. So you know that male gametophyte is non-motile, but the mobility is done with the tube nucleus, right? So tube nucleus form the tube till the first synergids. It will absorb the watery content from the synergids, and it will burst into the embryo sac, and it will release the non-motile motile male gametes into the embryo sac. Okay, so this release of or carrying the male gamete into the pollen tube is nothing but the sphenogamy. Okay, that the carrying of male gamete into the pollen tube and bursting into the embryo sac, that condition or the term for that is nothing but the sphenogamy, and that sphenogamy ensures the fertilization. Right? Next, syngamy and triple fusion I have written over here. Okay, syngamy is nothing but the fusion of male gamete. This green, hopefully you can see here that I have made a dot. Okay, it is the fusion. This is the male gamete fuses with the female gamete, that is the syngamy. And the triple fusion, these two are the secondary nucleus of the embryo sac or female gametophyte. It fuses with the male gametophyte. So it is 3N and it is 2N. Okay, so this, it is known as what? It is a syngamy, and it is what? Triple fusion, right? So now this will become, or this will restore the diploid condition, because you know that if male gametophyte is haploid and female gametophyte is haploid, once the fusion or syngamy happens, it will restore the diploid condition. So it will form the zygote, and from the triple fusion, that the secondary nucleus is there, it fuses with the another haploid male gametophyte, so it will become what? 3N. Okay, so it will produce the PEN, that is nothing but the primary endosperm nucleus from this triploid endosperm. Okay, and it will provide the nutrition for the developing zygote or developing embryo, you can call it, right? So one is what, from syngamy we will get, or the plant will get the zygote, and from this it will get the endosperm, that is primary endosperm nucleus. Okay, so it will provide the nutrient for the developing embryo, right? Hopefully you understand it. Now they have given the significance of this double fertilization, that it is a unique feature of angiosperm. It ensures that the parent plant invests a seed with the food store only if the egg is fertilized. The diploid zygote develop into an embryo, which consequently develops into a new plant. Next is what, the triploid PEN, that is primary endosperm nucleus, develop into the nutritive endosperm tissue. It restores the diploid condition by fusion of haploid male gamete with a haploid female gamete, that is a syngamy, and it also helps to avoid the polyembryony. Okay, so what is the polyembryony, we'll discuss in later, right? So these are the significance of double fertilization. It may ask you for the exam, for board exam. So double fertilization is very important. It comes for 3 or 5 marks, right? So do it well, student, so that you acquire this 3 or 5 marks, right? See you in the next lecture.